Thank you. It's kind of fun because I get to come to these conferences. This is, I think, my number 4, maybe. And every time I come, we talk about even more. How's that? Is that better? Every time I come to one of these conferences, I get to see all of you. I see a lot of familiar faces. And it's also just fun for not only for me, but also for my family. Because my family hears that I go to clinic and I see people, but it's not often that they get to come and they actually get to be with you. And so I actually appreciate very much the ability to come here with my kids and have them hang out with you as well, because that's a special thing for us. Today I'm going to tell a little bit about your story. In other words, the things I'm going to talk about have a lot to do with you, and many of you who have also joined not only my study, but also the studies of other individuals. It's also the stories of probably hundreds of other people who are not 1p36 patients or families. Because ultimately, these days, science is done kind of in a group all over the world. And so you're going to see that individuals from all kinds of different walks of life are helping out with things that could be of importance to your children. But let me just go ahead and we'll start first by saying, why study 1p36? Now, for some of you, this is really obvious. It's like, well, it's all about families, right? And these are our kids. And we recognize that, and that's probably the most important reason to study this. But there's also a lot of other people who have great interest in this as well. So, for example, doctors have interest in this. Now, I know many of you say, well, my doctor has never heard of 1p36. I'll tell you, if they're a clinical geneticist like me, and they have not heard of this disease, they are not going to do well on their board exams, because this is considered highly important, and because it affects, again, 1 out of every 4,000 individuals. And so this is something that we should know about. And if we don't know about it, then it's important that we get educated about that. But most physicians, when I go and we talk at places, they recognize this immediately, at least among the academic physicians. Also, we know that this causes significant medical problems, which is something we're interested in trying to help out with. Then there's scientists, and I'm a little bit of both. The scientists, they're particularly interested in learning about genes and also how things develop. And so studying this also gives us a chance to understand how different genes work and how the body develops. And so it's of interest actually more generally. And so again, when I talk to scientists, they're very interested in what we're learning about 1p36 as well. Now, if you said, well, what do we all want? If you said, well, what is everybody trying to get to do? In other words, what is the research all about? All of us really want to do 2 different things, at least. The first one is we want to know what are the medical problems that we should expect. And now sometimes you say, well, tell me what my child will do. That's a little bit hard. But at least on a more basic level, I want to know, both as a physician and a scientist and a parent, I want to know, what am I going to expect? What can I do? And are there things that I should be aware of that could happen to my child or I could avoid? The other thing is we want to actually be able to prevent and treat these medical problems. And so all of us are on board with that same thing. And if you ask scientists, they would say that's their purpose. And if you said physicians, they'd say that's their purpose, and that's your purpose probably too, right? And so I'm going to just talk a little bit about kind of not only how we're going to try to attack this, but specifically what are the challenges. One of the challenges is already spoken of by Scott, and he indicated that no 2 1p36 deletions are exactly the same. That's true. In fact, I'll guarantee you, if you have actually 2 children, well, you have 2 children in your family, they might have the same deletion. But if you take your child and you compare their deletion with anybody else's in this room, it will probably be different. In fact, I would bet money that it is different. Why? Because there's no specific common breakpoint for 1p36s. In other words, it doesn't happen over and over again the same way. And so that becomes a major challenge, because every child is going to be unique. And we can't just study everybody in general. We have to start to break individuals down into groups if we're really going to understand this. Just to give you a for instance, this is the 1p36 region, the top of chromosome 1. It's about 30 megabases, so it has about 30 million letters in it. And so if you say, well, this is a typical chromosome, one child, for example, might have a tip that's missing, and that might involve, let's say, 99 genes. Another child might have a bigger piece that's missing. That might involve 186 genes. Or you maybe your child doesn't have the tip that's missing, but instead has a piece that's in the middle that's missing. And again, that might be 77 genes. And so you can see that each individual may be different. The number of genes that's missing may be different. Even the kind of genes that are missing may be different. And so this becomes a little bit of a challenge. So not only that, but we also don't know the role of all those different genes. In a perfect world, we would know what every gene does during development and in adulthood, and then we would be able to backtrack and say, well, if that gene's missing, then maybe this would be the consequence. That's kind of what we're trying to go through. So this is the challenge, and the solution would be to make a map, and very specifically, kind of a gene deletion phenotype map. When we say phenotype, all that means is a medical problem. It's just a fancy word for saying that. But when you read things, you may hear that word, or your doctor may also say, well, that's a very interesting phenotype. All they're just saying is an interesting medical problem. This can also go into things like a way a child looks is a phenotype. Even the way the child acts can be a phenotype. But we want to know, again, what are the genes or the part of the chromosome that's leading to that phenotype? Now, this has been a problem that's been attacked for a long, long time. If you look on this list, this is a list of all the problems that typically happen or can happen for a child with 1p36 deletion. This is kind of considered the classic list. And so as a geneticist, I would learn this list, and I would know that those are the things I should be looking for. And if I see a child in clinic, then I'm going to be going through that list and saying, what are the problems that this particular child has? Well, for a long time, people have been trying to say, well, what part of chromosome 1 causes most of these problems? And so back in the 1990s, there was a group at Baylor, headed up by Lisa Schaefer, who has actually come and talked at these conferences before. And they said that based on their analysis, they could say that this distal region, distal meaning kind of far away from the center of the chromosome, this is a critical region that if you delete that piece, you can end up with all these different problems. Could end up with those. And so they said they think that most of the genes that are important in 1p36 are right up there at the top. Now you say, well, but my child doesn't have the tip that's missing. Well, as it turns out, there was another group from Baylor, which also in this time in 2007, found out that there's another critical region on chromosome 1, and guess what? It can cause almost the exact same list of problems. This was kind of unusual, unexpected. No one thought this would be the case. The children look slightly different, so the way their faces are maybe a little bit different, but the medical problems are exactly the same. And yet there's obviously two different sets of genes because these two regions don't overlap. And so we became interested in saying, well, if there's two critical regions, could you break those regions up even in more detail and be able to say exactly what regions cause each particular problem? And so that's something that we've been working on for now probably another almost 10 years. So if we just take, for example, that distal region, so this piece right here, which is called the classic region, and we broke it into pieces, and we said, well, what are the different phenotypes? We can show that having that open soft spot or fontanelle, which maybe your kids had for a very, very long time, that comes from this piece of the chromosome being missing. You can then say, well, how about hypothyroidism? Well, again, there's genes for that in this particular region. Cleft lip and palate. I actually don't see a lot of kids with cleft lip and palate in these conferences. Maybe, does anybody have a child that has cleft lip and palate? One over here. Okay, one over there. So a couple. This is a less common thing, but we can also, again, say that genes in this particular region are responsible for that. Seizures, lots of you, I'm afraid, have to deal with seizures. And so we actually have multiple different pieces of the chromosome that are important in seizures. That's probably because there's many different ways to cause problems with brain development. And so it's not unusual to think that there'd be more than one gene that would cause seizures, even in a small region. Hearing loss, the exact gene has not been identified yet, but we know that it's in this particular area. Congenital heart defects, this is something that I work on a lot. So I work on heart problems, holes in hearts. And we know that there's at least 2 small regions on the tip that are important for that. Cardiomyopathy, that's the problem with the heart muscle. And so we go through each one of these and we say, well, we can identify that particular area in this distal critical region. So that's making a fine map or fine mapping these phenotypes. The next thing we did is, so what we do in our lab is we've taken and done a massive literature review, and also we've recruited many of you to come into our studies, and we ask people to give us both clinical information and also the location of their deletions, and we make this big database. So right now we have about 200 patients or individuals that have isolated 1p36 deletions. In other words, they don't have any problems with any other chromosome. And then we have their clinical data, and by doing this, we can try to make smaller and smaller pieces and try to figure out what part controls what phenotypes or what medical problem. This is an example. So heart defects is something that, again, I said I work on. And so we took all the individuals in our database at the time, this was in 2014, and just listed them out and said, this is the deletions that they have. And then we looked for areas that are not overlapping. And so we can actually identify 5 different regions for heart defects on chromosome 1p36. So 1 here, 2, 3, 4, and 5. All based on individual patients that have heart defects. This is why any one child could hold the key to trying to determine what are the genes that cause this problem. And the best way to try to figure that out is to try to get as many children into our studies as we can, because we never know which one might have that key and help us to determine what the genes are. So we have these five regions, so we know that there has to be at least 5 genes for heart defects on 1p36. Those are those 5 regions. And then we say, well, what are the genes inside there that would make sense or might be the cause of those cardiac defects? To do that, we take both our database and then we also add to it molecular biology. What do I mean by that? Basically, this is just studying the way genes work, like in cells or where they're expressed at. And we also use animal models. And so by combining these with the regions, then we hopefully can figure out the genes that are important. As an example, let's say for heart defects, we would look at those different regions and we'd say, what are the genes inside those critical regions that are turned on during heart development? If a gene is not expressed or is not turned on in the heart, it's unlikely to be the cause of the heart defects. And so we can sometimes eliminate many different genes off our list just because they're not being expressed in the right place. But we do studies to try to figure that out. We also try to figure out how genes work together. And so if we know that a gene, for example, is in a pathway that causes problems with heart, and it's on 1p36 in a critical region, then it's much more likely that that's one of the genes that we should be looking at and studying more in detail. The other thing that we do is, I don't personally work on zebrafish, but there's many people who do, and there are some very good 1p36 researchers that use zebrafish. And zebrafish are a great model. They have hearts, they have eyes, they have all kinds of things that we have. And so they become very, very useful. And it's also very easy to manipulate genes in zebrafish. One of the harder models that is one that I work on, and I work on mostly on mice. And mice are extremely good for this because they have essentially the same type of heart that we all do. And many of their other features are very similar to humans, and so they become a great model. So going back to this, can we take molecular biology and animal models and say, what are the genes that are important for these heart defects? And the answer is we can identify sometimes one or sometimes even more genes for each one of these areas that are probably important in the development of cardiac defects. Do you begin to see how hard this is, though? Because again, we're not talking about one gene. We're talking about sometimes multiple genes that are in one area, and all of them may be working together to cause the heart defect. And so we want to study each one of these and then say, well, how does it work? How does it work together with other genes? And it becomes quite a complex puzzle. Anyway, doing this type of work, we also use a new technique. Not really a new technique, but some new technology that's helping us out a lot. This is just kind of say genomic technology. All of you know about genomic technology because probably your child was diagnosed using some of the new techniques that we have. When I say genomic technology, it's something that allows a doctor to look at all genes or all parts of chromosomes in one shot. Back when I first started to be a clinical geneticist, I would walk into the room, and if I could see your child and I knew what your child had, just by looking at them, I could do something. But if I wasn't sure, or if it wasn't clear, I could do nothing, because there was no way to look at things just generally. About 10 years ago, that began to change. And a lot of your kids have been diagnosed using a chromosome microarray analysis. That was one of the first genome-wide ways of searching for genetic problems. And so you've probably actually been the beneficiary of these kind of things, even if you didn't know that that was genomic technology. So for most of you, your children were diagnosed by getting a blood sample usually, and we took those white blood cells, extracted DNA, and then we would use basically a microarray, which is a little tiny glass slide. It's about that big. And on that, there will be hundreds to thousands of DNA probes, each one having a unique place in the genome. And so what that kind of looks like, if you look at 1p36, think about the chromosome and then think about just dotting it with all kinds of different little probes, each one representing a unique part of that chromosome. On most arrays, there'll be even many, many more probes than this. There would probably be 1,000 probes or more on just that one part of 1p36. We then use a laser scanner, which costs more than my house does. And you take that laser scanner and you put these chips in, and it scans them, and it comes out with a readout that looks, whoops, a little bit dark. Sorry, too dark. Up there somewhere in obscurity is an array. This is what the array looks like when it's blown up. But you can see that there are individual circles. Each one of those is printed out by a printer. It actually grows the DNA on the glass slide. You then take the DNA from a patient and you put it onto that, and it will light up in different colors. So there's usually green, yellow, and red, kind of like those spots. And if there's a green spot, it means that there's one copy of that area, which would mean a deletion, because most of us should have a copy from our mom and a copy from our dad. And so if a green would be a deletion, a yellow would be normal, and then a red would be a duplication or 3 copies. We can then take all that information with the laser scanner, put it through a computer, and we get readouts that look like this. And you can see that this is normal DNA, black, but this green patch, that's a deletion. And it can map it to a specific part on chromosome 1, and we can then blow this area up and look at it in detail, and I can actually define exactly where that deletion takes place. And this is how most of your kids were diagnosed. And so we actually, with the report that you get, we can say exactly what piece of DNA is missing, and it's probably unique to your child. In other words, there's probably no other child, perhaps in the entire world, that has the exact same deletion as your child does. In this particular case, this deletion is important because it's an autism deletion, and it has a very important gene called TSNAX, and we work on autism too. And so this is one of the arrays from one of the patients that was in one of our studies. You'll usually receive a report that looks like this. This one comes from the laboratory at Baylor, but pretty much any laboratory gives you the same kind of thing. The most important thing for us as a geneticist is this little box. It tells you where the deletion is, and it gives you very specific numbers that say basically down to within 100 letters or 1,000 letters, it'll tell us exactly where that deletion is. And because of that, we can backtrack and then say exactly what genes are missing in your child, which gives us great power. So this is a very valuable piece of paper to have. What's new? So there's a new technology that does not affect you perhaps as much as other families, but it's very important. And this is another genomic technology. In this particular case, we're looking we're not looking for big pieces of DNA that are missing. We're looking for actual letter changes. Now, we've been able to do this for a long time, but basically the idea is the same. We take white blood cells, we extract DNA, and then we sequence specific genes. In other words, we're actually looking at the letter code for individual genes. Now, we've been able to do this for a really long time, even longer since I've been a grad student. We were doing this. But what's powerful now is instead of doing one gene at a time, we can actually do all 20,000 plus genes in one shot. That test costs about $7,500 usually to run that on a clinical basis. But you know what? An echocardiogram might cost you $5,000. But this allows us to screen every single gene without even knowing what we're looking for. And then we give it to a laboratory, or the laboratory takes that information and says, I will look for changes in genes that can cause any of the problems that are on that list that the child has. This is extremely powerful because it's a way of looking blindly without even knowing and being able to identify what the problem might be in a child. That particular report usually is one of the longest ones we have. Often they're 7 pages long because they have all these different changes, 7 to 8 pages. And in this case, it will actually give a specific gene or specifically change in a gene, and it will say that's the gene that causes this problem in your child. The reason why that's powerful and important to you is that gives us the ability to not only understand what the deletion is doing, but the individual genes in the deletion. We're going to figure them out because we're going to learn about kids who have mutations in that gene. And so really these are like the more detailed level. If you want to think about them, they're like 1p36 deletion kids except for super small changes, super, super small. And so they're going to have specific medical problems, but they're going to be shared among your children as well. So with those techniques in place, then we can say, well, how do you actually do this? So let me give you 2 examples of how these stories play out. The first one is a story that's not mine. So this is a story that was done by another group. It has to do with a gene called PRDM16. No particular reason to know. All genes have names. Most of them mean absolutely nothing. PRDM16 is just a gene. But it's one that we're interested in because it's been shown to cause the cardiomyopathy that some of your children may have. And it's located in this distal critical region. And so the story goes like this. Basically, there were a whole bunch of families just like yours who had children with 1p36 deletions, and they joined various research studies, many different research studies. And that's kind of an important thing. And because of the many different research studies that they joined, we were able to access that information. We then have doctors who have then diagnosed some of these children with cardiomyopathy. And so actually working together with the clinical information and the diagnosis, and with your permission, then scientists took and analyzed all that data to try to find a causative gene. What does that look like? Well, in this particular case, what we do is we line up all the different deletions, or this group lined up all the different deletions of kids that had cardiomyopathy. And almost all of them lined up, and if you just kind of said, well, what do all of them have in common, except for this little one right over here? All of them had in common this one little section of DNA. What's in that section of DNA? As it turns out, it's only one gene, and that's the PRDM16 gene. This other person actually has a much bigger deletion that goes way over here onto the other screen somewhere. It actually has a different gene that's involved that's causing the cardiomyopathy for that child. But when the scientists saw that pattern, they said, we should look at this particular gene because it may be the one thing that's in common with all these patients. They said, well, does the gene make sense? So we say, well, the gene's a great candidate based on where it's at. But then they say, well, is it expressed in heart? And the answer is they looked at mice hearts, both during development. So this is a baby mouse before it's born, and an adult mouse. And although you can't see this very well, basically this gene was found in all of the cardiac tissues that usually have problems with cardiomyopathy. They then said, well, does it happen the same way in humans? And yes, fetal samples and adult samples of heart also have the same gene that's in them. So it's in the right place on the chromosome, it's in the right place in the mouse and in the humans. Then the question is, well, what about zebrafish? This particular group liked to work with zebrafish because they were easy to manipulate and they have good hearts that would be good for studying cardiomyopathy. Now, you don't know maybe what a zebrafish heart looks like, but it's a little bit different than ours. It has 2 chambers, not 4, but it looks kind of like a long sac-like thing like this. And they were able to do studies and say, you know what, the cells that are in there, in that heart, there's fewer cells than there's supposed to be. And so they'd use different ways of changing the gene, this PRDM16 gene in zebrafish, and they could say that not only were there not enough cells, but also the cells were not replicating or dividing as fast as they're supposed to be. They then went on to prove that there's all kinds of ways that that heart in zebrafish, when you knock down PRDM16, it didn't function correctly. It was very much the same way that it doesn't function correctly for kids that have cardiomyopathy. The next piece was probably the most important thing. They then took a whole bunch of non-1p36 deletion patients, individuals who had cardiomyopathy. So basically these are large cohorts of children and adults with cardiomyopathy, and said, well, if this gene's important for that, might we find changes in this gene in those individuals? And the answer is they did. This is a diagram of what the PRDM16 protein gene looks like. And they found 1, 2, 3, 4, 5, 6, 7 individuals in their group that had mutations in this gene. And they had the same types of cardiomyopathy that we often see in kids with 1p36. And so putting all this together, they can say, yes, right place on the chromosome, right place in the mice, right place in humans, right place in zebrafish, causes problems in zebrafish, and then we can even go back to the humans and say we have mutations in this gene that can cause this. This is done. Like, this is a complete story. And with this, we can have complete confidence that this gene is sufficient to cause cardiomyopathy for anyone who has it deleted. What does that mean? Well, so the next thing they do is they go ahead and they publish this. One of the things to look at, when you look at a publication, see how everybody has these little numbers behind their names? Those are all the different institutions that those people are part of. All you can do is it's just for fun. You count how many there are. There's 19. That means there's 19 different institutions that had people that went together and pulled together to make this paper, which is like, wow. You know, back in the day, you might have one place that did this. And today, no, we're pulling from all kinds of different places to make this research happen. So 19 different institutions and a ton of researchers all working to make this story happen. So what does that help? I'd say yes. The first thing to do is a geneticist can then sit down with your report on your child. And one of the questions they should ask is PRDM16 deleted? If PRDM16 is deleted, then your child would be at risk for cardiomyopathy. And then we say, well, okay, so what would we do about that? Well, one of the ways you can screen for cardiomyopathy is by an echocardiogram. And we talked a little bit about, well, how often do you screen kids with echocardiograms? A little bit unsure, but probably every 3 to 5 years, depending on your cardiologist, we should be screening a child that has a PRDM16 deletion. They should probably be followed by a cardiologist and have routine screening. That's a little different than we would do perhaps for all kids, but again, especially if that gene was missing, that would be important. What does the future hold? So I went on last night and just typed in PRDM16 research to see kind of what this group is up to, the group that discovered this. And one of the things is they're using both zebrafish and mouse models now to try to identify drugs that could change that. In other words, alter the levels. And they've actually had some success already using zebrafish so that there's some chemicals or some medications that you can use that would actually stop the problems with the heart in the zebrafish. And the idea then would be that could those same things ultimately be used for children and stop that particular problem from happening in kids with 1p36? If they can do that, it would not only benefit our kids, but also could benefit individuals who have that PRDM16 mutations that they were showing. And so again, this not only helps people in this room, but a much bigger circle of a whole bunch of other people who may have the problems with this, who may or may not know about 1p36, but are actually sharing the same medical problems that your kids are sharing. So that's story number one. Second story I'd like to share is one that we were part of, and it's kind of fun for us. This is a story that we've worked on for probably 10 years now. And it took us a long time to do this because we did not have the genomic technology to help us that much. But there's another gene called RERE, which I've actually talked about at other conferences, that causes multiple medical problems, and it's found in the proximal critical region. So not all of your children would have this particular gene missing, but for ones that take out this centerpiece, this could be a very important gene. This story happened in a little bit different way. We were interested in trying to determine what causes all the medical problems that are associated with this particular region, and we looked at all the different genes that were there. And of all the different genes, there was one that kind of stuck out especially to us, and that was RERE. And the reason why it was stuck out special is because we knew certain things about this gene. What it does in the body is it acts as what's called a nuclear receptor co-regulator. Now, what does that mean? Basically, it's a gene that makes a protein that interacts with other proteins, and then as a complex, they turn on other genes during development. And so we knew that this type of gene could interact with all kinds of different parts of your body. And because it was expressed in lots of different places, like the heart, brain, eyes, all these different areas, we said this could be a very, very important gene. We also knew that it was important in a thing called retinoic acid signaling. This particular signaling pathway is made up of a whole bunch of different genes, but it's especially important for heart development, brain development. So again, it was also the right kind of gene in the right place. So what do we do? Well, we went back to the same database. Now this, again, I should tell you that many of you are a part of that first study on PRDM16. Some of you may not even know that you're part of that. Why? Because we're constantly going back in the literature that's already been published and pulling all this information, right? And so this is all the different individuals who have RERE that's been deleted that, as far as we can tell, have ever been published. And so here's the RERE gene and all these individuals that are deleted for that gene. And then we say, well, what do all of these individuals have in common as far as their medical problems? And if you do that, it's basically the same list that we had for individuals that have that proximal deletion. And so we said, well, at least it's in the right place. The only thing that we didn't find was we could not find any individuals that had renal or kidney problems in this particular group. But this is helpful. So we're in the right place, interesting gene. The next big thing was that we got a mouse model. I'm a very religious person, and I don't think that things happen by chance. I will tell you that we received this mouse essentially completely by chance. There was a person who was working at with this mouse at Baylor College of Medicine. They were about to change jobs, and they approached me and said, Daryl, would you like to work on a mouse that has eye defects? I was not working on eye defects at the time. I said, can I think about it for a little while? I said, why give it to me though? Why don't you give it to somebody else? And he says, I've tried to give it to a bunch of different people and no one wants to work on this mouse. I was like, why? He goes, I have no idea. But if you'd be interested, I would give you this mouse and I'll give you the following data about that mouse. I said, okay. I was relatively naive, starting out my career. I said, sure, I'll take that mouse and we'll try to figure out what's causing the eye problems. Well, we got the mouse, and we figured out that this mouse had this change in the RERE gene. And we knew that it was important for eye defects because the mice don't have very good eyes. They have super small eyes. But that's not a typical finding for 1p36. But if you go back and look at that little tiny area, there are children that have microphthalmia or small eyes. They're in that area. But I knew about this gene, and so I said, I bet you this mouse has more going on than just that. And so we started to work down the list. And we said, okay, we know about the eye problems, right? But does it have postnatal growth retardation or deficiency? Basically, does the mouse grow after birth the way it's supposed to? And we found that when we tracked the weights of the mice, they started out the same weight, but then very quickly, the deficient mice just didn't gain weight the way they were supposed to. Then we said, well, could they have cognitive problems? So is there problems with their brain? We found out that their brains were smaller. We found out that the part of the brain that is especially involved in motor movements, it was abnormally formed. And we also found out that the special cells called Purkinje cells that are involved in, again, coordinating those movements, they're supposed to have these big spines on them that kind of look like trees. And when you looked at our mice, the trees were smaller and they didn't have as many branches, and so they weren't as developed as they were supposed to be. We then said, well, about clefting, and so we bred these mice onto a different background. At first, they did not have clefts, but on a different mouse background, we found that they have clefts. And so basically, this is kind of the tongue of the mouse. There should be this palate, and then this is the nose. And these guys that have their tongue and then their palate doesn't close correctly, and so you have basically a communication between the mouth and the nose, which is cleft palate. We checked their hearing. We found out that they have hearing loss. So this is kind of quiet noises, and this is loud noises. This is how a mouse is supposed to be able to hear. This is how our mice hear. So they have to hear really, really loud noises if they're going to hear anything. We looked at heart defects. We found all kinds of heart defects in our mice. Some of them you may have heard of, some of others not. Your aorta is supposed to come out of your heart and go around this side of your body. Their aortas go around the wrong side. They also hook up to the wrong side of their heart. So aorta is supposed to come out of the left side of the heart. It comes out of the right side of the heart in these mice, which is called double outlet right ventricle. We also found septal defects. Now this is probably one of the most common things. Probably a lot of your kids have atrial septal defects or ventricular septal defects. Our mice do too. And the other thing we found is that our mice also have cardiomyopathy. So if we let them live for a period of time and then look at their heart muscles, they have these big scars in their hearts. Not only do they have all these things, but when we looked at their kidneys, they also were missing their kidneys. So sometimes there would be mice that were born without any kidneys, and that's lethal in a mouse. And so this is 2 kidneys, and this is just 1 kidney for this mouse. The hard part for us, though, is that even though we had these mice for a very long time and we were convinced that this was a gene that was important, when you talk to other scientists and doctors, they said, well, that's interesting, but that's in mice. Can you prove it? In other words, can you prove that this gene is sufficient to cause all those medical problems? And I would have to answer over and over again. I'd say, well, no, I can't, because maybe it's some other genes that are in that same little area that are causing this problem. And they said, well, how would you prove it? I said, well, I've already done all the mouse studies that can be done. The only way you could really prove this is to find someone who has a mutation in that gene. And then that individual would tell us exactly what that gene does. And so we waited. The problem is that 3 or 4 years ago, no one was screening this gene. Why? Because we didn't have the technology to do that. And so it was only when you finally could screen all the genes at once that we had a chance. And as soon as that technique started to be used, we knew it was just a matter of time and we would find a child that had a mutation in RERE. And so we had to find that individual. And that individual was this child. He doesn't have 1p36, but he has something that's super important for 1p36. So he is the first child that was ever detected that had a mutation in RERE. His mom gave me a call. Why? Because she had read about 1p36 and all the stuff that we were doing about this gene. And she called me up and she said, I'm a mom of a young boy who has a mutation in this gene. It's not found in me or my spouse. It's a brand new change just in him. But we've read about your mice, and we think that our child has the same things your mice have. And so when we looked at the list, he had all these problems that our mice had too. And she said, I'm interested in trying to figure out if there's other parents that have this as well. I'm being seen at UCSF, but I wonder if there's other people out there too. In the end, we ended up I connected with her doctor at UCSF. He had already started to work on identifying other children that have this. Some of them did not have all these problems. He was a neurologist, but they would have something really simple like just autism. That's it. And yet there were other kids who had many, many more problems. But we began to work together and to try to find ways of finding more children. The way we did this was we would look through big databases. So there's a company called GeneDx that runs a lot of this genomic type of analysis. We found multiple patients through them and then contacted their physicians who then contacted those families and said, would you like to join this study? Because we want to know about this gene. We then also went on a thing called Gene Matcher. This is what I do. So there's a program that allows me as a physician or scientist to type in the name of a gene that I'm interested in. It then goes into a central area, and it's matched with everybody else who has interest in that gene. And then I get an email back that says, so-and-so is interested in this gene too. Then I write them an email, and I say, hey, I have the following interests. Do you have something that we could share? And so by doing that, we actually found individuals from all over the world physicians who had patients with changes in RERE, all of whom thought they may be one of the few or one of the only ones that had a patient like this. But when we started gathering them all together, we were able to find 10 different individuals, all of whom had brand new changes not seen in their parents in this particular gene. These are just kind of a schematic of how the gene looks, the different parts of the protein, and these are all the different changes that we found in those patients. What do we find? So there's the 10 patients that have the RERE mutations. They have all of these problems. If you then say, well, how do those compare to the problems with the kids with 1p36? There's a huge overlap. The only thing that's different is that these subjects didn't have cardiomyopathy, and the kids with 1p36 deletions did. Then we take a look at how does that compare to the mice. And again, all the things that are kind of in this middle section, all three of those different groups had those same problems. And so that includes brain abnormalities, eye defects, hearing loss, facial clefting or palatal clefting, congenital heart defects, growth deficiencies, all were in the same. And so basically we could say that, well, we can now say that RERE, just deleting that one gene, is sufficient to cause all these problems. Now that doesn't mean it's the only gene that can cause this, but it itself is enough to make that all happen. And so even if you have a teeny tiny deletion that just takes in that gene, you could end up with all the same type of characteristics that a child with 1p36 has. We then published our report, and once again, just to kind of look, we're going up to 15 on this, right? So 15 different institutions from all over the world, all coming together to make this one paper, this one thing happen. And it takes a lot of people to do this kind of research now. So how does that help? Well, the first thing is we know that if your child is deleted for RERE, there's a few things that should happen. And maybe you've probably already done this, but we would think about getting an ophthalmological exam because they can have abnormalities that sometimes cannot be seen on the inside part of their eyes. Echocardiograms would be important. Renal ultrasounds would also be important for these children, and especially they have problems with reflux. It seems like vesiculourethral reflux, basically when urine gets backed up into the kidneys and can cause damage. What does the future hold? Well, we have now zebrafish models and mouse models. We hold the mouse model. There's another group that works on zebrafish on this. And together we're working on all kinds of brain, eye, and heart studies, trying to figure out how this thing works. If you said, well, will this be easy to cure, or does this help us? One of the main challenges for us is that brain development happens way before a child is born, right? But there are some parts of it that continue on after birth. There are possibilities that we could begin to intervene with that piece, for example, and say, well, could you find a treatment that would help, for example, those Purkinje cells that control motor development, could we help them to grow more correctly? Eye, again, most of the development happens before. Heart, most development happens before a child's even born. These are hard things to change. But we would be interested in knowing how does that gene work. And I'll just say that as we learn how this gene works, we recognize that, again, you are not alone. So 1p36 heart problems, actually, it's the same pathway that causes problems on 8p23.1 deletions. You probably never heard of that, right? But guess what? There's almost as many of those kids as there are of your kids. And they have their own syndrome, and they want to know what's going on. They have a problem with a gene called GATA4, and it looks like RERE turns on GATA4. And so although you feel like you may be isolated, you're actually just one little spot on a huge web of a whole bunch of people who are interested in all these same genes. And they're meeting in their conferences talking about like who's going to help our kids. And the answer is you guys are. Why? Because all this is going to come together and we're going to solve a lot of problems. And in doing so, we're going to learn about a lot of different diseases, many of which have to do with 1p36 and many that don't. So how can we get more stories? This is finally get the last little piece done. How can we get more of these stories? So I told you only 2 stories, right? There's tons more stories to be told, I'll tell you. So one of the things you can do is you can sign up for 1p36 research studies. There's a bunch of them, but we would encourage you to sign up for those. If you'd like to be part of the database that we collect, we would love to have you because that does help us to figure out what genes might be important. This is my email address. You can email me. And I put that on there, and then I thought, you know what? It's probably easier in this day and age to text me, right? Everybody wants to just text me, and like, okay. So my phone number is 203-7242. You can text me and say, hey, I went to your talk. I'd be interested in joining. If you'll just text me that, you can probably give your name or your email address. Then we can get you in contact with Andres Hernandez, who's our clinical coordinator. He speaks Spanish, I speak Spanish, we all speak Spanish, because we're in Texas, right? I'm based in Houston. And so we would love to have you join our studies. And again, there's many other studies that we would encourage you to join the studies that are going to do good things. There's lots of them. But we would love to have you be part. The other thing is consider sponsoring 1p36 research studies. This was kind of talked about by Scott too. Research is very expensive. And one of the big challenges is, well, honestly, I spend most of my time writing grants, trying to get money to do the research studies that we want to do. Right now, we have probably on the order of 8 different papers that we are waiting to publish from our lab. Why? Because I'm too slow to get them written, and because I'm doing other things. That's just part of being a scientist these days. But if you have the ability to do so, you could consider, could I move this work forward in a much faster way? And the answer is yes. We just simply don't have enough people to do the work. And so if you have financial contributions or if you thought about fundraising, that would be great. I work on a lot of different disorders, and honestly, I work on the ones that we can get funding to do the work on, because we have to be able to pay everybody. This is where I live. So I work at Baylor College of Medicine. I work specifically at Texas Children's Hospital. That's where I have my clinic. A lot of people say, should I come visit you? Honestly, if you have a really good pediatrician or a really good clinical geneticist that's by you, they can do this. There's nothing special about my eyes, nothing special about the things that I know. I'm more than happy to email you and such, and we can talk. But honestly, you just need to find a really good person who's willing to get you plugged into all the places you need to be, right? This is my laboratory, so the people in red are the ones that work for me right now on 1p36, and the other 2 are ones that have graduated from my lab that have also worked on this project. We have our funding sources, which includes March of Dimes, American Heart. NIH has also sponsored us, which is National Institutes of Health. And probably more importantly than anything, thank you guys, because it's your kids and your willingness to help us out that makes the biggest difference. Because honestly, we wouldn't know where to even start if it wasn't for the help that we got from you. And so that I say thank you very much. And I know that my time's out, but if you have questions, I'm going to hang out for a little bit. Or you can email me or text me, and I'll try my best to answer you as fast as I can. Thank you so much.
Thank you so much, Dr. Scott.